Okay, so ladies and gentlemen, today my title is the lead to drug candidate, novel kinase inhibitor in cancer therapy. Currently, I'm working in the Institute of Biotechnology and Pharmaceutical Research, National Health Research Institute in Taiwan. So the IBPR mission is to establish, conduct, and support the research on new medicines and the biotechnology, and will lead to the better human health and the quality of life. So this is the IBPR pipeline, including the 4 different areas, such as the anti-cancer, anti-diabetes, and anti-infectious disease, and pain relief. Currently, we have 2 clinical candidates to reach almost the end of the phase 1 clinical trial. And then currently also have the 5 development candidates are undergo the preclinical development. Due to the time limit, today I only can focus on the DBPR 114 as the multiple kinase inhibitor. So what's the protein kinase? There are more than 500 protein kinase encoding in the human genome. So the kinase is regulate signal transduction pathway in the cell, and then play the important role in cell growth, metabolism, and apoptosis. Since the Gleevec was launched in 2001, the kinase are the second most sought after the drug target. In the next 2 slides, I show you currently there are 27 kinase inhibitors in the market. So this first is 15. As you can see, the blue is indicated in 2011. 8 kinase was approved by the FDA, and then 2012 they have the 4 was approved by FDA. Last year they have the 3. This year already have the 2 approved by the FDA. So among of the 27 kinase inhibitor, you can classify from the very selective lapartinib into the multi-target kinases such as the sorafenib and the sunitinib. So you look at how multi-kinase can earn a big revenue. For example, sorafenib can be treated advanced RCC and hepatoma carcinoma. Sunitinib can treat the patient with the RCC, GIST, and the PNET. Both drugs have more than 1 billion revenue per year. So that we want to develop the multiple kinase inhibitor. So how we develop at the NHI? Well, we start with the Aurora kinase. Aurora kinase are the mitotic kinase for cell division and are overexpressed in the number of the human cancer cells. So if you want interested in this area, I already wrote the 3 review papers from the expert opinion. So how we identify the 1K724 as the Aurora kinase inhibitor? We utilize the 4 different platform technology. Okay, first is the knowledge-based screening. Second is the structure-based design. Third is the high-throughput parallel synthesis. Fourth is the lead optimization. So when we start this project, first we use the virtual screening to get the 1K25 as the Aurora kinase. At that time, the IC50 is around 15 micromolar. So after we synthesize more than 75 analog, we improve the potency 354. And then we solve the A compound as the X-ray co-crystal structure. So that is and then we published this result in Journal Medicinal Chemistry in 2009. So, but however, this is the compound have some stability problem. So we changed another strategy called knowledge-based screening to identify the BPR1K224. So when you look the substrate, when you look at the ATP binding pocket, you will see this is a hydrogen donor, is a hydrogen acceptor. They have the two regions. One is the hydrophobic region 1 and the hydrophobic region 2. Then one day my postdoc came to my office and said, hey, Dr. Hsieh, I think we can find the kinase inhibitor just by the drawing. I asked him, so show me how to do that. So when we draw the structure by the IC space, and then we define that they have the hydrogen bond donor and the hydrogen bond acceptor, something like that. And then we screen our in-house library, more than 125,000 compounds. Then we get around the 3,000 compounds to fulfill this sub-search. After we do the second search, we get the 62 compound fulfill the 2 round the screening. And then we test the 21 compound for the aurora kinase activity to obtain the compound 7 with the IC50 3.8 micromolar. As the medicinal chemist, you will quickly recognize, oh, this is useful, skillful. So we do the substructural search and then to get another 16 compound. And finally, we identify the compound A is the 224 as around the 300 nanomolar. So it means we only test 37 compound. 21 plus the 16 together the compound, one compound with the nanomolar potency. So that is the 224. Then we go for the structure-based drug design. My colleague, Dr. Wu Suying, to get the Aurora A crystal, and then to make the 224 as the co-crystal. Once you solve the co-crystal, you found that this is the empty hole on the back pocket. So then we want to extend our chemical structure. So by using the high throughput parallel synthesis. So what means the HTPS? We make the 350 kinase focus library. As you can see, this is our key intermediate rare with the different amines. So, and then we just do the assay without the purification. Once the active compound show the 90% inhibition at the 10 micromolar, and we resynthesize our compound. So you can see it's the 224, and then we got another compound called is the 430, but both compounds still have the good IC50 enzyme activity, but do not have the cell-based activity. So once we found that we have another NH2, we make the urea to get the compound 432 with less than the 50 nanomolar, and then the cell-based activity show 400 nanomolar. So then we do the co-crystal structure again. So you can see the urea part will go into the back pocket, and then this nitrogen is oxygen combined with the gatekeeper. So they improve the potency. But however, you have the good enzyme activity. That doesn't mean you have they can show the in vivo efficacy. So the first 32 do the standard in vivo activity, did not show any activity. So what's wrong? Since we need to improve the lead property to more favorable the drug-like properties. So then finally we use the lead optimization, 3 optimization strategy were improved. Now, the optimization based on the structural property relationship information. So you look in the 432, the C log P is 7.4, and then the molecular weight is pretty high. So we need to reduce the molecular weight and then introduce the solubilized group to make the compound more drug-like, or based on the Lipinski, the rule of 5. So we do the intensive the optimization for the 3 strategy, see this side and scale 4 and another pharmacophore. So finally, we identify the DPPR 114 as the multiple kinase. As you can see, 724 is the 3rd generation. The 114 is 4th generation. And after the 45 fix the kinase profiling, you can see the DPPR 114 have is really multiple kinase at the 1 micromolar, more than 77 kinase was show the good activity. Okay, so now we examine the in vivo activity. The first is MV411 AML xenograph. You can see the 1 milligram here, and the 3 milligram, and the 10 milligram during the 10 days the treatment. So after stop the treatment, you can see the tumor won't come back. So the second in vivo model is the pancreatin, the MIA-kappa-2, the pancreatin. So you can see the 20 mpk. When we give the 20 mpk in IV, the tumor is shrink, not only delay growth, but the shrink the tumor. The third model is hepatoma. Hepatoma is a very difficult model to treat. So compare, we use the sorafenib and our compound on 10 mpk and the 20 mpk. So the compound can show the good activity at the 20 mpk. So once we identify the 114 as the drug candidate, the first we go the source selection, because the compound has the solubilized group. We do the 2 round the screening. One is the test the 100 screening condition, and then we choose the 8 hit or identify. Finally, we got the softform A and the softform B. As you can see, this table is this free form. And this soft form on soft form A is the hydrate, high crystalline. So B is the unhydrated, but it's the medium crystalline. But for the solubility, you can see the form A and show the good, much improved water solubility than the free form. So now we choose the form B as the preclinical candidate. So you can see from the DSC and the TGA and then PLM is the crystalline, and then this is the data. So I summarize currently the status of the DPPR 114. How we identify the last year, currently we undergo the API production, and this year Q3 we'll start the rat and the dog the toxicology. Hopefully we will file the IND in the Q2 of 2017. So we identified, we already synthesized 12,000 compounds from the BPR1K series. We more than 100 compounds evaluated for acute and PK study. The 20 compound evaluated in the in vivo xenograph to identify the DPPR114 to show in the in vivo efficacy in pancreatic cancer, hepatoma, gastric, bladder, colorectal, and then AML. So we already published the 12 publication, and the 5 patent was granted. So that is the 3 recent publication from the Journal Medicinal Chemistry, and also we published in PNS 2 years ago. The last year we published in the ChemMaking as the cover story. So we also get a series of the patent status. So before I let you go, I should appreciate our technology, our collaborator. I'm led by the medicinal chemistry lab, the bioassay lab, led by my colleague, the structural biology, high throughput lab, the animal pharmacology, and the pharmacokinetics lab. So I also thank my group. Without their tireless effort, I won't stand here to share our experience. So I have the first slide to entertain everybody. People may know Taiwan or may not. Okay, the Taiwan is for the Portugal word is the Formosa. It means a beautiful island. So the Taiwan is near the mainland China. Is they have the subway, okay? And then the area is 3600. It's probably the nine times larger than the Dubai. Okay, I do check the Dubai from the wiki, and then the population is 20,000 million people. So what's the best thing? Scenario view. Okay, so if you have the chance, welcome to visit Taiwan, the Alishan National Park, Takonga National Park, the Salmon Lake, or Yushan Mountain. However, we do have another one. It's called Taipei 101. Before the Kilometer Tower was established in Dubai, the Taiwan 101 is the tallest building in the world. So if next time you have the chance, you should visit Taipei 101 to go to the top and see the whole Taipei city. Finally, I thank for everybody to attend the meeting, and also thank the organizer and the section chair to give me the opportunity to share our result. Thank you for your attention.
Thank you. Do you have any question?
Excellent.
Thank you.
Nobody. Thank you.
The certificate.
Please, Mr. The certificate.